Good morning, everyone. My name is A Ramani, and I will be covering ion channels and neural communications today. Today we will be covering these objectives. Describe the purpose of the nervous system, explain signal transduction, discuss the differences between a ligand and a voltage ion channel, explore membrane potentials, and review channelopathies. First, I would like to start with our nervous system. Your nervous system is your body's command center. It is composed of the brain, the spinal cord, and the network of nerves. Our nervous system uses neurons, which are our network of nerves to send messages back and forth from our brain to the spinal cord and to the nerves throughout our body. As humans, we use words and body language to communicate. Neurons use electrical and chemical signals. Examples of neural functions include a knee-jerk reflex or even making a decision. We have billions of neurons. Every day, our brain processes millions of bits of information. And in response, we are able to carry out thousands of different acts. Each neuron is connected to thousands of other neurons. They communicate through electrical and chemical signal. Each neuron has the same basic structure. The cell body is located right here. Then you have the nucleus, which contains the genetic material. You have dendrites, which receive information from other neurons. Then you have axons that send information down to its multiple terminals, also known as the terminal button. Most synapses communicate through chemical messengers. Other synapses are electrical. In these synapses, ions flow directly between cells. Typically, chemical transmission involves the release of chemical messengers called neurotransmitters. Neurotransmitters will carry information from the presynaptic or sending neuron to the postsynaptic, the receiving neuron. So looking at this visual, the presynaptic or sending neuron is right here. This causes a transmission of a signal or neurotransmitter to the other neuron, the postsynaptic, which is located right here. So this is how the transmission of the signal occurs, and this is how neurons communicate with each other. As we receive and interpret these signals in the form of light, heat, odors, touch, or sound, we react and respond to it. But before the communication can take place between neurons, there is a series of electrical changes that must occur within the neurons. Many signal transduction pathways amplify signals, and this leads to the activation of other molecules. Examples include neurotransmitters, hormones, and cytokines. Cytokines help control inflammation in the body. Proteins that detect and convert these stimuli through chemical, physical, or electrical signals are called receptors. Many signal transduction pathways require the binding of a signaling molecule to the receptor, which ends up triggering events inside a cell. The binding causes a change in the receptor, which leads to a cellular response. So how does this happen? Embedded in the surface of cell membranes are protein structures called ion channels. Here are a few examples of the ion channels. They are pores or holes in the lipid bilayer. They act like gates to allow or stop the flow of ions in and out of the cells. For transport efficiency, Channels have an advantage over carrier protein transport substances in that up to 100 million ions can pass through one open channel each second. Ion channel proteins transport substances only down a concentration gradient. Carrier protein transport substances can travel down as well as against the concentration gradient. Ion channels are usually divided into two categories: ligand-gated channels or voltage-gated channels. As we talked about earlier, each neuron holds an electrical charge. In a resting state, the charge is about minus 70 millivolts, and you can see in this diagram here it shows minus 70 millivolts. So basically at rest there is a negative charge. In neurons, potassium ions are maintained at a high concentration within the cell. So the within the cell is shown here at the bottom, while the sodium ions are maintained at a high concentration outside of the cell. So you'll see here the extracellular fluid. As we talked about in the last slide, ion channel proteins transport substances down a concentration gradient. The cell has potassium and sodium leakage channels, and I will describe that further in the next slide, that allow the two ions to diffuse down their concentration gradient. This means a molecule will move from a higher concentration to a lower concentration. So you see here inside the cell, potassium is in a higher quantity versus outside the cell, sodium is. So since sodium is at a higher concentration outside of the cell, it will tend to move into the cell. Potassium is more concentrated inside the cell, so it will tend to move outside of the cell. So this makes the inside of the cell slightly negatively charged when compared to the outside. These channels can open if the right message is received from the neighboring neuron. I wanted to take a step back and talk about leakage channels, which I brought up in the last slide. If you notice here along the lipid bilayer, there are a variety of color-coded ion channels. The blue dotted channels are sodium leak channels. The potassium is striped green, and the chloride is yellow. You can see here that the neurons have far more potassium leakage channels than sodium channels. So you notice that the blue dotted, there's a couple there, but there's 1, 2, 3, 4, 5 striped green leakage channels. At rest, the membrane is the most permeable to potassium, and this leads to potassium's outward flow. The membrane is also permeable to chloride ions as well as sodium ions. So you may wonder, if the cell has open, non-gated ion channels and ions were moving at rest, won't the cell reach a point where potassium is at a state of equilibrium if it is only permeable to potassium? We're going to answer that in the next slide. So I'm going to focus on the sodium and potassium channels here. In this slide, you will hopefully be able to better see that there are more potassium non-gated ion channels when compared to sodium channels, which are here shown in a circled in blue versus the green ones are circled in red. If only the non-gated potassium channels were open, then the membrane potential would reach potassium's equilibrium potential, because as you see, there are so many more potassium striped green ion channels. But if you notice, there's also other channels available, such as the chloride and the sodium. But there are a fewer number of these channels compared to potassium. So this causes the inside of the cell to be more negatively charged than the outside of the cell. These actions are maintained by the sodium-potassium pump, which helps maintain this resting potential. The sodium-potassium pump is responsible for maintaining the electrochemical gradients needed for neuron functioning. To the previous slide, here are the dendrites. So that's where the neuron will receive the signal. The ion channels, as a result, will open on the neural membrane. Then, because of this change in charge and concentration differences with the sodium ions, the internal charge of the cell becomes more positive, as you can see here. As the cell's charge becomes more positive or less negative, it is called depolarization. This alters the ability of specific types of ions to enter or exit the cell. So remembering in our last slide, when a cell charge becomes more positive or less negative, it is called depolarization. If the charge reaches a certain level, called the threshold of excitation, neurons become more active and an action potential begins. So many additional pores will open, causing a massive influx of sodium ions, which you can see here, and a huge positive spike in the membrane potential, which is called the peak action potential. At the peak of the spike, the sodium gates close and the potassium gates will open. As positively charged potassium ions leave, the cell begins repolarization. At the peak action potential, potassium channels will open and the cell will become hyperpolarized. At first, it becomes slightly more negative than the resting potential, and then it levels off, returning back to its resting potential. So what is our resting potential again? It's when our sodium channels will return back to their resting state and will be ready to open again. And then our extra potassium ions will diffuse out of the cells through the potassium leakage channels, bringing the cell from its hyperpolarized state back to its resting membrane potential. This graph gives a nice summary. So if you remember, in the resting state, it's about -70 millivolts. So thinking about this, in the neurons, the potassium ions are maintained at a high concentration within the cell, while the sodium ions are maintained at a high concentration outside of the cells. And so that gives its negative charge. Now, depolarization occurs around here, and that's where the neuron receives a signal and will suddenly change. This is where signals that are received from one neuron to the other occurs. The ion channels will open on the neural membrane. At this point, you will see the sodium ions will be propelled into the cell, With this influx of positive ions, the internal charge of the cell becomes more positive. As you see, it's beginning to become more positive. If this charge reaches a certain level, the neuron will become active and an action potential begins. This is what is called the threshold of excitation. So again, you notice it's becoming less negative. So many additional pores will open, causing a massive influx of sodium ions, and a huge positive spike in the membrane potential occurs, and you'll reach this peak action potential, and the membrane is definitely positive at this point. So at the peak action, the sodium gates will close and the potassium gates will open. As positively charged potassium ions leave and head back to the cells, the cell begins to repolarize. So you will notice it will hyperpolarize first, which means it becomes even more negative than what it was at its resting potential. But then it will head back towards resting potential, which means that our sodium channels will return to their resting state, and the majority of them will close and be ready to open again, and then the extra potassium ions will diffuse out of the cell through the potassium leakage channels, bringing the cell from its hyperpolarized state back to its resting membrane potential. So this process can happen a couple of different ways. Together, the combined effort of ligand-gated and voltage-gated ion channels are involved in heart and skeletal muscle contraction, for example. When it comes to memory and cognition, the central nervous system is involved. The voltage-gated ion channels only let one kind of ion in, while the ligand-gated channels are less selective and allow two or more types of ions to pass through the channel pores. And in the next few slides, we're going to dive into this. Ligand-gated channels are opened by a neurotransmitter that fits into the receptor site like a key into a lock, as you can see here. The signaling chemical known as a ligand binds to the receptor molecule, which causes a cellular or tissue response, including a change in the cell's electrical activity. Neurotransmitters such as serotonin or dopamine will bind to this specific receptor site on the ligand-gated ion channel. This triggers a modification in the structure, and this activation allows the inward or outward movement of certain ions. This leads to changes in membrane potential and cellular responses. Voltage-gated channels are where two neurons are close enough together so that the electrical charge from one neuron can travel to the next neuron. The opening and closing of these channels may depend on the binding of the signaling molecules such as neurotransmitters from the ligand-gated ion channels or on the voltage across the membrane. For example, the voltage-gated ion channels. As we discussed earlier, neurons can produce electrical activity through voltage potentials by the unequal distribution and movement of ions, which are electrically charged atoms across the neuron cell membrane. I'm just wanting to give you a little bit more of a visual about what I talked about in the last slide concerning the voltage-gated channels. Again, they're normally where there are two neurons close enough together so that the electrical charge from one neuron can travel to the next. So Here we see a nerve impulse beginning at the presynaptic or sending cell. To release the neurotransmitter, the action potential is then converted at the synapse here, or the gap between two neurons. After that, the postsynaptic cell, which is across the synapse, will react to that neurotransmitter. Now we know how ion channels and neurons are supposed to function and communicate, but what if they do not? Channelopathies are a group of genetic, autoimmune, or inflammatory conditions that result from the dysfunction of ion channels, which are located in the membranes of all cells. The most frequent cause of channelopathies are mutations in the genes that encode ion channels, which we know will affect ion channel function. Ion channel defects have been implicated in a variety of diseases and conditions, including epilepsy, migraines, blindness, deafness, diabetes, high blood pressure, asthma, irritable bowel, and even cancer. As you can see, it affects multiple organ systems. A specific example of this are pain channelopathies. They are a class of neurological disorders. There are a wide array of ion channels that are expressed in the nociceptive neurons. These neurons detect and alert the brain of imminent danger or injury, but deficits in these ion channels have been found. They are associated with defective pain pathways. Episodic or extreme pain syndrome usually begins in childhood or infancy, and they're known to result from gain of function mutations of a voltage-gated sodium channel that causes abnormal electrical firing. So neurons in the end are hyperexcitable. A gain of function mutation is a genetic lesion that causes a normal part of the gene to be expressed inappropriately, or at abnormally high levels, or at the wrong time. So in extreme pain disorder, it results in a hyperactive sodium channel. It does not close properly. So this lengthens the action potential and increases the intensity of pain signals downstream. There are pain-relieving medications that are now targeting potassium and sodium ion channels because of this disorder. So let's think about it. In these situations, you want to regulate neural excitability. So you would want to focus on the ion potassium channels, which are key regulators for setting that resting membrane potential, repolarization, and then limiting the excitability of the neurons. Sodium ion channels are also another focus for medication, because when you think about it, the membrane depolarizes above the threshold of excitation. And in that moment, there's that influx of sodium ions, which initiates that action potential. So that in the end can lead to pain signaling. Now, the opposite can also occur. They can experience a loss of function mutation, which leads to an congenital indifference to pain. There are non-functional units in the sodium channels, so they are not able to open properly. So this prevents the flow of sodium ions into the nociceptors. This lack of sodium ion influx blocks nociceptors from transmitting pain signals from the site of an injury to the brain. Unfortunately, there is no treatment for this. So you can see why proper functioning of ion channels are so important. So continued, deeper understanding of the structure and the function of ion channels can help researchers develop medications that can help provide treatment for these and many other diseases and conditions related to ion channels. Thank you, and are there any questions?